So welcome back. We are still here at the EFIC TV studio in Budapest. It's sort of late, 3:20 on the second day. People are a bit worn, but here in the studio with me I have two people who will make one heck of an interview, I'm very sure of that. This is what we expect, so a nice conversation about something that is quite technical, to be honest. Because I know you just look like sweet people who dropped in and wanted to chat about tea, but essentially what you are is really skilled technicians and highly trained experts in your fields. I would like you to introduce yourselves, so maybe you go first. So you are titled with something that we're not going to call you, so your real name is, I'm going to read it, Shafaq Sikandar,
Yes,
is it?
that's correct.
And you are actually Danish, which I am as well.
Yep.
But you do live in England, so.
Yes, so I moved to the UK almost 20 years ago for my undergraduate degree. Happened to do a great project in the lab that I ended up doing a PhD in, and that's where I met Kirsty. So
was that the UCL?
That was at UCL in the pharmacology department, and so it's a real pleasure to be sitting here with Kirsty here almost 20 years
But
later,
we're not introducing Kirsty yet, we want to introduce
you. Oh no,
you.
but she's very important. So.
She is, I agree, yeah. So now, and you are just recently appointed professor?
Yes, I was I'm very delighted. I feel very lucky and fortunate to be in this position. Less than 5 years ago, I got my faculty position at Queen Mary University of London at the William Harvey Research Institutes. The institute directors took a gamble on me. I tried my best, and I have a really lovely little group there, and we do some preclinical research and some clinical studies in pain mechanisms. So we're very much focused on musculoskeletal pain at the moment.
Nice. We'll come back to that. So, yeah. Kirsty, who on earth are you?
Sorry,
Kirsty Bannister.
Yeah, so hi again. Who am I? So I'm original member of the Sulky Corner, which is what we manifested when I joined Tony Dickinson's group. Our corner was allowed to drink a lot of tea, always smelled of nail varnish, and we were allowed to sulk quite a lot when experiments went wrong. And actually, so Skip was doing a PhD and I came to Tony's lab to do a postdoc with no pain and no neuroscience. So although Skip was the PhD student, probably for the first few years Skip was teaching me everything. That is the truth of it. And we bonded over the cryostat, brain slicing, etc. And so, yeah, who I am today, I want it to be in the context of who Skip is, especially because we're celebrating a professorship, which is just, the most incredible and deserved recognition of amazing work for an amazing person who, first and foremost, is one of my best friends. So when the invite came through to do this with Skip, it's like this,
I would just like to say that when I was told by Kirsty that we're doing this together, sorry, it took me about 48 hours to clock that. I was very happy.
So Skip thought, because I was telling her about this interview.
So you just go ahead and talk, and I'll talk to the people
sorry.
out there. You see how easy my job is?
I actually forgot we were being recorded. Okay, sorry.
No, it's fine.
Ready for the questions?
Yeah.
Okay, let's do questions then. So it feels like, what is that, a talk show host? I don't know. Yeah.
Rain or sin. You're just going to have to rain or sin.
Well, but this is what, I mean, before we go into it, I think this is really important. Obviously, if you're not here, maybe you have been to a conference, maybe you haven't, but I think it's important for people to realize also that one of the reasons that people like us go to conferences is because of this. Because you need to meet people and connect, and so obviously you live in close proximity, but you meet people here that you don't speak to every so often. But whenever you come back, it's that same network and it's that same atmosphere, which is what we talked about before. So let's keep it in that atmosphere, so that it not only behind the camera, but also in front of camera, because that's what research is as well, isn't it?
Yeah.
And I'll take a stand. So my training was a physio, so I have this clinical background, and we always talk about patients. but neither of you have that clinical aspect on it, yet you are both doing research that hopefully now and in the future will benefit patients. And the sort of theme we're talking about here is how do you do research in rodents that are obviously distinct from humans and translate it into something useful that can help me, the clinician in me at least, understand what is going on. So that's the big topic. And what I would like you to start talking about, because that was when I started doing basic research, that was one of the really eye-opening things, is that this is not just something you fumble into. You really train yourself to do this. And I imagine doing, how big is the spinal cord or the brain of a mouse, I'm imagining, right? So, and you need to stick something in or do something and find that right spot. How do you do that? It's just, I don't know. How do you go about doing that?
I mean, the training element is long-winded. When you're learning to do a spinal nerve ligation, you're doing it on cadavers for quite some time.
So spinal nerve ligation means you take out the sciatic nerve and then you bind a little bit of thread around it.
So not that you take it out, but you expose the spinal cord and you find the thread of the nerve that you want to ligate, so you're going to tie it tightly. But that's the kind of surgical procedure that you have to be really quite proficient in a dead rat or mouse before you're let loose on the anaesthetized live animal. So I suppose we're very dexterous, I think it's fair to say, because we're working with very small parts.
No, absolutely. And I think a lot of this is I think in any walk of life, if you want to do your job well, you just have to keep practicing, training until you're spot on 100%. And even having said that, Kirsty's probably witnessed me many times in the lab having opened up a rat or a mouse spinal cord and trying to do a dorsal horn recording, and I totally fail. I cannot find a cell for the life of me. And even when you do have that technical proficiency, there's an element of luck to it as well, so it's about resilience as well.
And how do you know you're in the right spot?
So you mean the receptive field of where we record from is the hind paw. So while you're looking for the cell you want, which as electrophysiologists, it's about what you can hear. You can hear the cell, right, that you're looking for. It makes a certain noise.
Receptive field means a piece of the skin on the mouse. On the leg, for instance.
So the nerves that are innervating the part of the cord that we've got our recording electrode in, their free endings are on the toes of the hind paw. And there'd be days, so we would sit back to back at the electrophysiology rigs, and there would be days when your first insertion of the electrode, you would find a cell. That's a party moment. Right.
That's sweet. That's pretty sweet.
It doesn't happen often, but when it happens, you're going to make sure everyone knows about it, and you're going to be really smart, because there can be other days where you're 4-5 hours tapping your receptive field, and the electrodes are taking it in and out, and moving the depths, and you don't get anywhere. And those days are hard. That's when we would have a lot of chocolate. Skip would bring out the Danish licorice, which no one else liked.
Salty licorice all the way.
Absolutely, I'm with you on that one.
Disgusting. But it's whatever it takes to get ourselves through.
So what are the tools you need? I mean, are you using microscopes? Can you do it with your bare eyes?
I mean, I think it depends on the animal that you're using. Rats.
So let's say you want to do a dorsal root recording. So you're doing something to the sensory part of the efferent, the sensory efferent.
Yeah.
How do you know? So you listen for the right spot, but I mean,
you physically know that you would be in the lumbar part of the dorsal horn, and even for example in my lab we also do dorsal root ganglia recordings. I mean, you can actually count which ganglia you're
and you
in.
can see them. They're big enough for you to see, so they'll be on the size of.
I mean, sometimes you can use one of those those head-bearing microscopes. But if your eyes are better than mine, you don't need them. I have terrible eyesight. But rats are a bit bigger, so obviously a little bit easier to work with just by virtue of them being larger. Also, they're much cuter than mine.
I mean, how do you, obviously you don't make the tools, but how are the tools made? They have to be so thin, I imagine, to record from a single
really
cell.
Standard tools that are available from a lot of companies, and they're miniature versions of what you might find in a theater, which is a little bit weird.
Yeah.
So if you ever walk into a hospital theater and you recognize rongeurs, you're like, wait. But you know, they are effectively tools that a lot of orthopedic surgeons might use as well.
Okay.
And they're just fine forceps, fine scissors.
And everyone is like really picky about their rongeurs. So each rig would have their own rongeurs, and God help you if you walked in and red rig rongeurs were on my blue rig, because I'd be like, who has got my rongeurs? And it's like, it becomes like voodoo, and you have to be using your rongeurs. It becomes quite personal, doesn't it?
Yeah.
In terms of that equipment you're using, and you like this kind of curve, and you like that kind of curve, and it does become. There's a bit of magic to electrophysiology, and there's a lot of luck, and there's a little bit of voodoo.
Yeah, I think tools are very personal.
Yeah.
I mean, I have actually, if I ever throw all my toys out of the pram, it's because someone's taken my Watchman forceps.
Exactly.
And so I look under
Yeah.
the microscope and one tip is broken. I'm pretty sure it wasn't me.
Stay out of Skip's way at that moment.
Yeah, definitely stay out of my way. I will have a proper freak out.
Yeah, no, that's bad.
But it does speak to what we talked about in the beginning, that this is not, you know, a game that you just play. It's not just random and you stick it in and whatever happens. And I mean, even in humans, it's really difficult. So just look at the size difference, right? Which also begs the next question. So how can we use the information we get there? Like even action potentials, and I know this is very random for you, but let's just start at a very random point. So is a C fiber similar? Are the voltage gated channels, are they the same?
I mean, I guess if you're recording from the spinal cord, then you're recording from quite likely second order neurons.
Yeah.
So these neurons will be receiving a lot of this primary afferent barrage. There'll be several types of fiber types that are synapsing with that second order neuron that you're recording from. If you're recording from the dorsal root ganglia, you can then record from individual, or at least at the level of the soma, you can actually record from individual primary afferent fibers firing, and based on conduction velocity, or even just the same way we characterize WDR neurons in the dorsal horn, you get a better idea of whether they're mechanospecific, thermospecific, or wide dynamic range type of neurons. So,
and there was this work done by Cellular Lawson, a while back, where she looked at the differences between.
Yes.
So
A-betas.
we keep saying that if it
okay.
had this velocity, or if it had these capacities, or if it had these receptors, then we call them something specific. And it turns out that depending on what criteria you use, you could be talking about the same cell, but you give it different characteristics or classifications. She said there would be like a 20% overlap between A delta fibers and C fibers.
Yeah,
So
and
how
a
do you
lot of
know
work
what
on
you're
A-beta
working on?
fibers, A-beta nociceptors, very important to point out. Everyone talks about A-betas as if they're like these innocent little children. No, they're bad boys. They're
And when you
important for pain.
define them as A-betas, is that based on velocity? So the speed they conduct action potential with?
Yes.
Yeah. Would it be so? What about the size of them?
Could be
Yeah.
heavily myelinated, yes.
Yeah.
But we in the in vivo ephys prep, you wouldn't necessarily be looking at myelination property, because you'd be recording.
Because when
at
you're
the level
doing
of the soma.
electrophysiology, what you get is the action potential. So the
It's the latency.
Yeah.
So we separate it post hoc histogram by latency in terms of A beta, A delta, C post discharge. I mean, the program does it for you, so it's quite neat, right?
So what if you wanted to know what kind of receptors was on this specific cell? How would you do that? That would be a different method, right?
You can use genetically encoded calcium indicators with transgenic mice, and then you can actually visualize the cells. So the beauty of ephys is that you get quite a lot of.
So that was a really foreign word for me.
Oh, okay.
Ephys.
Electrophysiology.
Oh,
so
okay.
you got to get in the game, man. Shortcuts. No, so I mean, the beauty of electrophysiology is that you get a lot of information about the graded coding of cells. With calcium indicators, again, you get information about the activity of neurons. Calcium transients aren't important for action potentials, sodium flux is. But obviously there are calcium transients when neurons are activated. The nice thing about using something like calcium imaging, which is, some people do calcium imaging and electrophysiology at the same time. That's pretty wild, but it's cool. The nice thing about using calcium imaging is that you can actually use reporter mice. So you might have all sensory neurons in green, but there might be a TD tomato reporter for a particular subset of neurons. It might be a subset of neurons that express a sodium channel subtype. We often use NaV 1.8 as a reporter when we're doing
so that's
calcium
a
imaging.
voltage gated sodium channel, and it's called 1.8 because that's what it's called.
And so you can say, okay, I know that NaV 1.8 neurons respond to these stimuli and non-NaV 1.8 neurons don't, and you can answer different questions that way.
And I guess what I'm sort of coming to here is that there's also, even though you're very specific, highly trained, you only see part of the picture, don't you? So you never see, we do beautiful drawings or schematics, and it's almost like you see everything. You have a C fiber, you have these receptors and these kinds of channels, but that's not the real world, is it? You have to piece piece pieces of the puzzle together and say, I know this, therefore it has to be.
Yeah, there's a lot of leaps of faith between what you're seeing even when you do GCamp recordings. The penetration of depth isn't so high in terms of knowing precisely what your fiber density at any one point is, right? And again, you're going to determine what fiber type, for example, by the diameter. But actually, there are so many places where we're piecing the puzzle together blind, but we're doing the best we can with the tools we've got, and the tools we've got are actually really effective.
Have you ever come across an idea where everyone is saying that this is, whenever you see this, you can conclude this, but then it turns out you were wrong? Have you ever
all
seen
the
it?
time.
I was going to say, quite often.
Most of the things I test my hypothesis for are pretty wrong. So, which is great, and that's the beauty of science, right? That's what makes it fun, is that you can be pleasantly surprised, and it takes your work on a different tangent. And that's how you make really nice, new, exciting discoveries as well, by finding things you didn't expect to find.
And also it's that acceptance, I think, of it is what it is. You get a result and it might not have been what you were hoping for, but hey, this is what it is. So yeah, let's build a new path to walk along to explore that. I mean, I think for most of us, that is the really exciting part of research.
And I think this really hypothesis-driven idea, where you test a hypothesis and you're true to your results, not your theory, is, I had a chat earlier today with clinicians, so Tash and Romy, as you know, and we talked about that's probably one of the things that we should be much better at as clinicians. So talking about translation, I actually think there's something about the like being rigorous and sticking to your methods and knowing your stuff and then applying it and seeing what happens, not believing that just because it's supposed to happen, it actually does, but really being cautious about what you see and what you conclude.
And really critical as well. I think it's really important that we criticise our own work and we take great steps to convince ourselves of what we're seeing. We might do some dirty pharmacology, but we want to see there's another experimental approach with a different manipulation to give us the same answer. And I think there's a lot of that in the work we do too.
Yeah, absolutely. And I think just generally to get that big paper out, you do have to show the same thing in using multiple approaches, right? So electrophysiology is one approach, then you might turn to imaging, then you might turn to spatial profiling, you might go crazy.
Throw in a bit of behavior.
Yeah.
If you're really feeling crazy.
Is that when you act behavior, is that when you say now they're patients?
I mean, the behavior aspect for me is actually the worst part of the experimental approach.
Oh, but they're so cute sometimes.
I mean, rats in particular, right? Rats are so friendly and they recognize your smell and they get excited when you walk in the room, whereas mice are just bitey and smelly, and I don't like mice as much as rats.
I don't like mice as much as rats, but I think just because we have to work with them more, they can be really cute, but they are bitey.
They're so bitey.
I mean, you can work at them, work with them at your own risk.
Yeah.
Yeah.
Talk about cultural differences, right? I mean, I never had that discussion with any of my colleagues.
Oh, really?
So,
The bitey patient.
no,
that
the
comes
bitey
in.
patient
Yeah,
and
yeah.
no, the one who knows me by my smell. No, never had that with my.
But
You
it's
might, you just don't
interesting.
know.
Or
about
maybe
it.
I should have.
But it's interesting you
right.
draw this parallel because even with the patient work we do, we think of animal behavior and with rodent behavior, people that can do rodent behavior pain assays are amazing.
Yeah.
They have the patience. That's unbelievable.
Yeah, I agree.
But actually, that same sort of patience, compassion, and understanding is really important when working with patients that have chronic pain as well.
Yeah.
So you might be applying very similar stimuli when you're profiling pain in patients, von Frey hairs or like
Pinprick.
mechanical prods,
Yeah.
things like that. The same sort of stimuli we use in animals, but you have to be very patient when it comes to working with patient cohorts, especially when someone is in a lot of pain and they've taken out time to come to your clinic to then get tested and poked and prodded when they're already in this hypersensitized state. So there are some parallels.
I think we already covered that. When I imagine you would see patients, you both see or do research on patients as well. So when you do that, you'll be using the same rigor, different tools obviously, and different methods, different designs, but you'll be translating what you see on the bench, as it were, to the bedside.
Well, you say different tools, but one of the main aims in my group is that we are using completely parallel paradigms with completely parallel tools and eliciting stimuli.
But you're not spinal ligating your patients.
No. We're not doing spinal ligations, but in terms of the psychophysical protocols we apply to induce, for example, a condition pain modulation paradigm, temporal summation of pain, wind up, offset analgesia, onset hyperalgesia, we can use the identical paradigm, the identical stimulating tools in animals.
And if anybody would be interested in knowing those paradigms, you have an article coming out too, actually, on that?
Yes, so we've got, one of them is on BioArchive. It's under final review. I won't say where in case we get dumped at final review. And then the other half of that data isn't in paper form yet. We're still collecting the data. So yeah, I mean, it's a bit like what we did with DNIC in CPM, and we did the cuff algometry paradigms and showed that you can use an identical approach.
Yeah. So what you did there is in human models we use this cuff. So we do one painful stimulus, and we rate it, and we do this CPM paradigm, as we call it, and we can see that a painful stimulus or noxious stimulus can make another painful stimulus feel less painful.
Yeah.
And we do that with cuffs, and exactly what you say, you build then a tiny miniature version of that
neonatal blood pressure cuff. Yeah.
And you put that on a mouse or rat?
This was rat. Mine's too small for how big the cuffs are. And then we use the identical, the average conditioning stimulus pressure is 70 kilopascal. And so we were using that as our conditioning stimulus while we were applying the exact same test stimulus. So like really marrying and obtaining a spinal correlate from the anesthetized animal to what the perceptual correlate is in terms of pain rating on the NRS.
Yeah, and the beauty of that is that obviously you both work with Anthony Dickens and we had DJ here before who worked with Lebas, and they sort of showed this DNIC, the diffuse noxious inhibitory controls, that is a really established model of assessing the descending modulatory, or part of the descending modulatory pain system. And then we have the cuff system, which we think or thought was a parallel to that, and you actually showed that it is, because you could see some parallels.
Well, it's definitely the case that when you apply the parallel paradigm, the DNIC circuitry is being activated in the humans, but the percept from that condition pain modulation paradigm is driven by many other factors as well.
So what you're saying is that DNIC is very specific, and the DNIC parts, as it were, or pathways as we would call it, are activated, but CPM, the paradigm in humans, is different but overlapping.
It's underpinned by activity in the DNIC circuitry, but these people are awake. So there's all kinds of cortical influences that aren't there in the anesthetized rat. There'll be propriospinal mechanisms as well. Yeah. But we can be sure that one of the underpinning mechanisms of the condition pain modulation paradigm is activity in the DNIC circuit.
And this is, I think, where things get really interesting, because what you are doing in anesthetized rats, or even parts of a rat, even on neurons sometimes, is of course looking at it from a perspective of, I was going to say, measuring in both ends. You know what comes in, you know what comes out. And you keep it very simple and reductionistic, as it's sometimes called. So you have this problem, you reduce it to a single thing that you can actually study, so you know when you're wrong. And then we start applying it to humans, which are obviously so much more, being alive and having a conscious experience of the world is so much more complicated.
Yeah.
So how do we marry that?
I mean, there's different, so like Kirsty's approach of using identical stimulation parameters across species is a really good one, because actually in ways like a lot of those the like brain centers and nervous system centers that are involved in processing pain have been evolutionarily conserved across species. Totally works. Another approach that we try to do in the lab is to actually use patient samples and test how they affect neuronal activity in rodents. Now, it might not work for all cells and all molecules because there might be cross-species activation or with the immune system, but actually we've shown that like you can actually take cells from patients who have chronic pain and just inject them into naive recipient mice and induce a transient pain state in those mice.
So the cell that is living, that is real in vivo, as well, in this person, if you take that out and implant it into an animal, the animal will have behaviors that are believed to be representing pain. Is that what you're saying?
Yes, but obviously very specifically neutrophils from patients with fibromyalgia. So well, like not any cell.
But again, this is, I guess this is, and you know, this is how you need to do. You can't just do randomly and say, I think this is what happened. You need to measure it, right? So that's the premise of doing basic research, is you need to be reductionistic. But if you take neutrophils from a patient with fibromyalgia and put those neutrophils from a living person into a mouse, that mouse is going to have behaviors that simulate pain?
Yeah, so that mouse will exhibit evoke pain behavior. The spinal cord neurons and DRG neurons in that mouse are going to start firing away. And if you take out the dorsal root ganglia, you're going to actually see that those human neutrophils have actually started to infiltrate the ganglia as well.
And what will happen over time? Because you said it's transient. So what will happen over time in the mouse? Will its immune defense sort of?
Well, neutrophils are quite short-lived cells in general. So usually within a few days, the life cycle would. That's how long the life cycle would be. In certain diseases, the life cycle can be a little bit longer. So those neutrophils get cleared. So you can actually do whole body imaging of neutrophils if you tag them with a radioisotope, and you see that like most of them get like stuck in the lung. And then a lot of them go around into different organs, and a very small proportion, but a significant proportion, will make contact with nervous tissue, so with the dorsal root ganglia, and then they get cleared.
Okay, so they're not carrying, as it were, pathology or a disease state.
The cells are pathological.
Yeah.
They have some sort of pathology. Now, how they actually sensitize neurons, we don't know. We don't know if it's direct cell-cell contact, or the neutrophils basically have the job of getting close to sensory neurons, and then they release something that then...
What if you took a neutrophil from me, I don't have fibromyalgia, and put it into a mouse? Wouldn't it respond the same way?
Zilch.
No.
Zilch. I mean, if you don't have pain, mouse will be fine.
I mean, if I didn't know better, I would think that was interesting.
I hope so. I hope someone's reading the papers we publish. I don't know.
So what would be the perspective of this? Again, so I guess what we've established that it's not just guessing. It's really hardcore measuring. And we can do translational even at a cellular level, or even at, taking from human methods or paradigms into animals. So what is there a limitation to this? Again, coming back, maybe is the human the sort of homeostasis, for lack of a better term, or allostasis, for lack of a better term? Is it too complex? Will the body always return to a state or proceed to the next stage? Or could we put a single cell or a combination of cells into an animal and create what we know as a human condition that will be in that animal, not just as long as the cells are there?
I mean, I think that the tricky thing is that we're trying to model pain in rodents, and pain itself is a cortical experience, right? So, and a mouse cortex or a rat cortex is not the same as a human cortex, which is why I think we've strategically stuck to subcortical regions when we do our research, because those are regions that are evolutionarily conserved across species, and we can infer about pain mechanisms there. But how a human might report pain, where it's a combination of all of that affective and sensory input, is very different to what a mouse might be perceiving. We wouldn't know because a mouse can't talk and tell us what they're experiencing.
And I guess if they could, they would be likely to experience pain in a different way, again, maybe based on the differences in nervous systems anyway. So, but I think the big picture here is we want to use animal research to understand humans, and then we want to test treatments or whatever in animals to see if they would affect humans. But it's interesting that we can also take things from our world and put it into the animal world and test if what we are doing in the clinic or in the laboratories is really transferable. But where does this end? I mean...
Well, yeah, and I think that's the tricky part, right? Because translation more and more for the kind of work we're doing is going backwards. It's going from the patient domain to the animals, where we're trying to understand circuitry, function, anatomy, pharmacology, doing the kind of techniques Skip's outlined, using experimental approaches where we're just trying to say, does a paradigm evoke something similar in an animal that it does in a human? That's then going to inform how we're going forward, right, in terms of our forward translation experiments. But your question of where does it end, I don't think it does end. I think it just is an ongoing cycle, which sometimes has to, more and more I think it's the backwards leap that is going to be more effective.
This is I know exactly that you have a picture in your head, but there's a lot of backwards and forwards and fingers going around the air. So what if we talk about Parkinson's? What would it mean?
So we now know that Parkinson's patients can be absolutely stratified into cohorts according to a sensory phenotype, and that when you do that, there is a certain cohort of Parkinson's patient who has a neurogenergic phenotype. This phenotype is largely based on non-motor symptoms, sensory phenotyping in terms of sensory profiles, but also dysautonomia, sleep REM, and other more sympathetic nervous system phenotypes. Now, what you want to now do is you've identified a cohort, you've stratified a cohort. You need to now backwards translate that to the bench and use that information to generate a meaningful model of Parkinson's, which is driven by a neurogenergic phenotype. So to do that, what you're going to go after? The locus coeruleus, because that's generating 95% of the central nervous system's
So the locus coeruleus is part of the spinal cord, so it will be...
Not necessarily. So you've obviously
in the CNS, sorry.
Yeah, so you've got that output from the brainstem to the dorsal horn. We know about that sensory pathway, but you've also got connections with thalamic nuclei regulating things like tremor, the PAG, the hypothalamus, cortical brain regions that are regulating reward processing, etc. So in doing that, in taking what we've understood about stratified patient cohorts in Parkinson's in patients, right, it's meaningful data, we're doing that as we speak, and backwards translating that and saying, here is an animal model that finally represents one such stratified cohort from the clinic. And to do that, it's about generating a neurogenergic model of Parkinson's, where we're talking about a collection of syndromes rather than a single disease entity.
Because once you have isolated a phenotype in an animal, then that opens up for this avenue of doing what we can do in animal research, but we can't do in human research.
Exactly, and then that is where I think you get the really meaningful data from for forward translation to the clinic. But until we've understood what the clinical phenotype is, the cohort stratification, and we've generated the right animal models, how well can we then move from animal model back forward to the clinic?
And whilst Parkinson's is a disease that is identifiable and there are characteristics that goes beyond the sensory system as well, what about chronic pain? Where are we? What would be the ideal? What would you like to contribute to in the world of chronic musculoskeletal pain?
Blue sky thinking.
Oh God. Well, I think one thing that we probably haven't done well in the pain field is to really avoid this idea of just thinking of chronic pain as a single entity and thinking that there are sort of just homogeneous, these mechanisms that exist across different disease states. The approach we take in our lab is to really study different disease states independently and study disease within those pain states. So cancer pain or chemotherapy-evoked pain is not the same as bone cancer pain. It's not the same as pain in rheumatoid arthritis. It's not the same as pain in lupus. There might be some common mechanisms, but if we, from the get-go, think that there's a shared mechanism across all diseases, we're pretty likely to fail. So this is where that patient stratification is really important, because even within something like rheumatoid arthritis, actually we know already now that there's different synovial histopathotypes based on the different immune cells that infiltrate that diseased joint.
So the way the joint looks chemically is different in different people.
Exactly, and they might all be diagnosed with rheumatoid arthritis. They might all have the same DAS28 scores, so clinically they might all look the same, but actually the pathology is very different. The mechanisms of pain are quite likely to be different, just logically, right? The mechanisms of pain are quite likely to be different. Maybe some of the central modulation is the same, but the peripheral drive will be different. So we really need to try and understand. Like Kirsty said, I totally agree. Better patient stratification is so important for us to be able to understand which are the best animal models to use to reproduce and recapitulate even just aspects of human disease and pain. Because when we use fibromyalgia models in the lab, no, our mice don't necessarily have sleep deprivation.
So when you say fibromyalgia, is that a mouse model?
Yeah, so a mouse model of fibromyalgia in the lab, no, it's not sleep deprivation, cognitive fog. We're looking at sensory dysfunction in this mouse. That's it. So I'm very reticent to say that we study fibromyalgia in mice, but we use mouse models to study an aspect of fibromyalgia disease. So, and I think Kirsty's done this a lot, and I really hope, I can see from the posters here, a lot of people are using this very focused approach of looking at specific mechanisms, specific aspects of disease and pain dysfunction. And I think the more specific, and I guess, it's a bit like being picky, right? But the more specific we are, I think the closer we'll get to actually being able to put different pieces of the puzzle together. It
also places this reductionistic approach in the holistic model of pain, doesn't it? Because we know we're not studying the whole thing. Go 20 years back, maybe we thought differently, but now we know that we are not. But it's still legitimate to focus on a single thing, because there is legitimacy to whatever, a mechanism-based model, as it were. So I think that's really important. But what do you want to fix?
Because I've gone into clinic and I've talked to patients, I think that's been quite it's been an eye-opener, but a really good experience for me, because I think when I started my training as a scientist, I hadn't really spoken to patients that had chronic pain, like any kind of pain from any disease. And it's only by being more engaged with this sort of translational focus of my work, I actually really understand. Well, I think I understand, or I try to understand, the problems patients experience. What is their biggest problem? How is pain affecting their lives? And it really isn't that much effort, actually, to go and speak to patients, try and understand what's important for them and work that into your applications, the questions that you're asking in your research. I think it's been a really fruitful experience for me. So what I would want is just to have impact, positive impact, for patients that suffer from chronic pain.
That's almost like
disease.
A contemporary quote of Pat Wall, isn't it? So in order to do good basic research, you need to see patients to sort of put it into context.
I mean, while recognising that as academics, our impact on patient-centric outcomes is limited, but it's way less limited than it was 10 years ago. And I think one of the main drivers actually for change has been the fact that now you can't submit a research grant that includes patient populations without the patient voice. You got to have patients there from the beginning, even when you're generating your hypothesis. Are you, as a patient group, are you interested in this? Is this a question you care about? Yeah. What do you think about these tests? Would you be happy to come to the clinic and sit and have these applied? And I think it's really forced our generation of scientists to classify ourselves as basic science researchers, yet work in that clinical domain. It's really forced us to incorporate what matters to the patient, and I think that actually has really enriched our research approaches, and it's impacted how much impact we can have on patient-centric outcomes. So I've noticed a massive shift in that over the last decades, for the better.
And so in terms of being enriched, I can just say I've definitely been enriched through our talk here. It's been an absolute pleasure. Don't know if you want to come out with last comments or ideas or thoughts?
I mean, now I'm just thinking about salty licorice
Yeah.
and nail varnish, so probably no.
Well, I mean, I think another thing is it's great. You know, I think talking about patient impact is really important, but actually even the journey of being a scientist is just so rewarding. And I think about 20 years of friendship between us, and even on a personal level, like there's just so many reasons why we should love our jobs. Like, and I love my job because of moments like this, because I get to spend time with one of my best friends, but I also have that hope that the work that I do in the lab, through meeting people like Kirsty, can actually have a positive impact on people that suffer from chronic pain.
Amen.
Let that be the last words. Other than I want to thank you for your time. It's been an absolute pleasure and very enlightening. Thank you so much.